Hello, today I just want to talk about animal biotechnology and give you an introduction to it and make you see it through the perspective of our original question when we started this course. You remember I asked you about basic research and whether it was important or not. At the beginning of this course, we talked about spider silk and whether it's important to understand basic things like how spiders live or how do they make their silk, such basic research. That was the first question I asked you. So, in this lecture, I want to highlight spider silk research as an illustrative example, showing you how it is that profound biotechnological innovations are attempted to be made using this fundamental biological knowledge. So, the whole point of that is to make you understand from this example that animal biotech starts usually with some kind of basic research, and then the possibilities are up to you, the researcher. So, what is animal biotech? A quick definition is the use of modern biotechnological tools and techniques to improve animal health, productivity, and utility. Excuse me, just bear with me, I'm not that well. And examples of this are genetic engineering, cloning, genome editing, and even reproductive technologies. If you look at these three things here, genetic engineering, cloning, and genome editing, I want you to ask yourself the question, are they talking about exactly the same thing? Or is it just different words for the same thing? Or are they different things altogether? So genome editing is a new tool that's come on the scene, but genetic engineering, cloning, we've been doing that for quite a while. So your job is to understand the nuances between genetic engineering, which was the beginning, cloning, and genome editing, using the modern CRISPR tools and such, and also reproductive technologies. So what we're doing with basic research, so this is where the story begins, because you can't have biotech without the people who are doing the basic research to start with. When you're doing basic research, you're seeking to understand how the biological mechanisms work. How does life work? How do the processes work, the fine structure? What happens? You're just trying to find out. You don't get pressed by immediate financial goals. You're not looking for a commercial output here. When you're doing basic or fundamental research, you're looking for answers. You're asking why. Why do things work the way they do? And so, because when people do the research in this way, they're free from financial implications, they're free to actually just investigate how things really work. Then, once they've found that out, that is when the application questions come up. So, for example, studying the molecular structure and genes responsible for spider silk proteins, MASP1 and MASP2, understanding their extraordinary strength and elasticity. So you can have a project where you're just looking at the extraordinary strength of these proteins, the elasticity of these proteins. You can delve much deeper, go to the molecular level. So you may look at the actual sequence. Can you look at the primary structure, the secondary structure, the tertiary structure, the quaternary structure? Can you get insights as to what makes these proteins so special? Because function follows form. You always have the form first, and that's what determines what the function is going to be. I think I got that the right way around. But in terms of biological things, you have the sequence, and if you follow why the sequence is like that, the primary sequence, you follow why certain amino acids are incorporated into this protein, what does that contribute to the final conformation of the protein, and then how does that contribute to its function? So you always find the answers right there in the primary structure. So to illustrate that, I want you to explain how spider silk is produced and to provide one faster sequence of a spider silk protein. You know where to get this data, so please find it. You'll notice in this presentation, I don't have any images because I don't like spiders. So even in yours, please just put the FASTA, put the explanation. We don't need images. Thank you. You can submit this by the end of term at 17 hours, and you'll do it online through a portal, which I will share. So why is basic research vital? It unlocks new knowledge about biology, and then that is what you use to harness innovation. So the animal biotech comes and is built off the back of those who've done the knowledge work in the beginning. I want that to get through so that you never find yourself in a discussion where you're thinking which one is more important, basic or applied. They're both important. They're both two sides of the same coin. So basic research also provides the foundation for developing new methods and technologies. And it may initially seem that pure science is kind of blue sky. You don't know what on earth you're looking for, but it usually leads to very unexpected applications. So the person who does the research probably won't be the same person who comes up with the application. Basic research deciphered the genetic code of silk proteins and their mechanical properties. It revealed how they can be synthesized recombinantly by inserting spider silk genes into mammalian animals. And then, on the other side, you had to also understand how do mammary glands work. Because in this setup, these proteins were going to be expressed heterogeneously, not homogeneously. So they weren't going to be expressed in the same organism that they were being made. They were coming from spider, and then they're going to be introduced into another mammal, and then the protein was going to be secreted in the milk, meaning the mammary gland. So you need somebody who's understanding mammary gland biology. They develop the techniques so that you know exactly how mammary glands work. Then you can target your protein to be expressed in the milk, and you have this protein available now in amounts that you couldn't manage if you're trying to get them from spiders. So this is how this works. So you can see here you've got the basic researchers and the applied researchers. So how do you transition from the basic to applied research? Here's your application in animal biotech. So we use the insights from basic research to develop practical applications, products, or technologies. An example of this is creating transgenic goats that produce spider proteins in their milk. Then you go and you harvest the milk, and in the milk you find your biotech product, which is the spider silk. The process of applied research in spider silk example, you have engineered goats, so you have to use molecular tools, and then you have to go down to what's in the milk, the actual structure of the milk and the proteins in there. So how do you target the spider protein to actually, how do you target the spider silk to be produced only in the mammary gland and not somewhere else? You need to have a promoter which is specific to the tissue that you want. So in this case, it's a beta-casein promoter. This drives the silk protein expression in the mammary gland. So it's not going to be expressed in the eye of the goat or in some other tissue, to be expressed in the mammary gland. Then you've got to overcome challenges such as yields. So these are the biggest challenges of animal biotech, because you have to remember that when this protein is being expressed in its native host, it's being expressed for a purpose. It's not being expressed for commercial applications, right? So it's usually low yield, low amounts. So you have to do the biotech to increase the yields, and then you've got to figure out how you purify this protein. Once you get it from the milk, how do you take it away from all the other proteins that are in there so you can collect your protein? And then how do you do that while you maintain the protein properties? So this means that a lot of animal biotech is about optimizing production methods, possibly using gene editing tools that are available now so that you can improve the expression levels of your protein. These were not available all that time ago, but now you can use these tools to try to increase your yield. Applied research in animal biotechnology more broadly. So what other things can be done? You can develop gene editing animals resistant to disease. You can create pharmaceuticals such as A-trin. This is produced in genetically modified goats. And you can also have advances in cloning and assisted reproductive technologies. So we haven't talked much about the reproductive technologies, but this example here just shows you what is possible. So the take home here is that you need both types of research for sustainable innovation. You can't have one without the other. You need the people who do the basic research to provide the moving parts, then the applied people put them together to create something that is useful to society. So basic research discoveries often take years, and it takes a long time to find these things out. It might be 2, 3, 4 PhDs who are all working one after the other on a specific topic before the breakthrough comes out. Then once the breakthrough is there, that's when the applied researcher would turn it into something. Applied research bridges the gap between lab discoveries and marketable biotech products, addressing practical problems such as food security, animal health, and environmental sustainability. And these are really crucial for our situation here in Africa. So this technology is super important for us. I want you to think about the ethical considerations which have to be made when you're doing this kind of work. You should also look for the regulatory frameworks that are available in Zambia for actually regulating this. And I would like you, in this question, what ethical considerations arise in this study? I would like you to consider the National Biosafety Authority and its newly published regulatory framework on gene editing. So this is out this year, and I expect you to download it or at least find some information about it and know what is allowed now in Zambia concerning gene editing. So this is a very new piece of legislature that has come through in Zambia. So I'd love for you to know about it. And if I ask you what it is, you should at least have an idea. Then finally, we have the whole entire research pipeline. So how does the whole thing fit together? If you look at spider silk production, it's a brilliant example of how fundamental biology, that's basic research on spider silk genes and protein properties, how that enabled the innovation in the biotech applications. So that is applied research in transgenic animals. So when you look at this, I want you to think, even as a biotechnologist, you can choose. You don't have to be the one working with the animals. You can be the one who's just investigating how these proteins are. You can be a bioinformatician who's looking at the bioinformatics to help predict why proteins behave the way they do. You can be involved at any stage of this whole process. So you can be there at the very beginning where you're asking fundamental questions. Then you can be there in the end where you're asking, what can I do with this? You can also be in the ethics, because for you to do bioethics, you need to understand the technology in the first place. So you can also train in bioethics and be the person who's sort of like the umpire in this, the umpire, who makes sure that things are done properly. It is important to have a mindset that values curiosity-driven science coupled with problem-solving research approaches. Both research types are essential to push the frontiers of animal biotechnology to improve livestock and to contribute to food security and health worldwide. Sorry. So you should have some thought about the things that we've talked about here, but also think about these three questions, because one of them, I'll put them in the test. What are the other examples of basic research that led to unexpected biotechnological breakthroughs? How can applied animal biotechnology address challenges faced by African agriculture? And the ethics, what ethical issues arise when you move from basic to applied biotech? So if you look at this whole topic and consider it in this way, you should have at least a good picture of what animal biotech is, why it's important, why we need it, the different aspects of it where you can join the conversation. You don't have to be right at the end where you're dealing with the transgenic animals. You can be, if the whole thing makes you feel terrible for your own reasons, religious or philosophical, you might be into the ethics. So when you hear that there's a science like this might push you to understand the ethics and become involved in it in that way. So I hope that you're understanding what we mean by animal biotech, how the different types of technologies differ, and that there's also a legislature which is available right now in Zambia to manage this particular technology. You should also consult the table of technologies that is on the online portal, so that you can see the 5 technologies that were first introduced from Animal Biotech, and the 5 which are the latest ones, which are there, to give you an idea of how the science has come. Thank you.